Hi, my name is Brycen. I'm going to help share about and talk about DNA replication. And this helps kind of break it down into some simple pieces. DNA replication can be very, very complicated, but hopefully this will help break it down. This is what we're going to start with. There's one molecule of DNA, some DNA nucleotides and enzymes, things like RNA polymerase, DNA polymerase, things like that. So to start, something I'm going to help share is just a part of the DNA. So we have your phosphates and we have your sugars. And we have your bases. The bases always go on the sugar side. And that will be, this is what I want to remember, is the 5' and the 3'. Because that direction of the DNA is going to be an important step here in a minute. So on the opposite side, because DNA has the two sides, we've got our bases and we've got our sugars and our phosphates. And they match up just a little bit differently. Now, the bonds that hold all of these together, between these bonds, they're called covalent bonds. And the bonds that hold these ones together are called hydrogen bonds. So, on this side, you're going to be going down this way, and then this one is going up this way. And what we're talking about is replication, where we're going to separate these two strands. We're going to break this hydrogen bond. The two strands are going to separate, and then we're going to add nucleotides. Once we have those molecules, we're going to take the DNA, and I'm just going to draw it out really quick. Now, like we talked about a little bit earlier, there's those hydrogen bonds that are keeping the DNA together, and so what we need is for those to separate. First thing, there's something called gyrase and topoisomerase. What those do is they kind of relax the DNA, and then there comes in called helicase right here, and what that does is it separates the strands, kind of unzips them. So once the helicase unzips them, what this part right here is it's called the replication fork, and that's just simply where the DNA is separating. So this is stage one. We have our DNA, we have the helicase that is breaking those bonds, that's unzipping it, and this right here is a replication fork. So all of that is stage one. So at stage two, what we're going to do is we're going to synthesize the leading strand. What the leading strand means is that we're going to add the nucleotides to the 3' end. And so we're going to take this prime end right here. This is our leading strand. So an enzyme called DNA polymerase comes in and attaches all of them together, and it comes, and that one happens pretty easily. The new strand is going to go from the 5' end to the 3' end, and it's just going to go that way. To recap from the beginning, this is stage two. For stage one, we learned about the helicase, the replication fork. On this side, we're synthesizing the leading strand. We have our DNA polymerase, adding those nucleotides to that 3' end right there, and then now it's going this way. We have a whole new molecule of DNA. Stage three, we're going to synthesize the lagging strand, and so with this one, it's a little bit different than the leading strand. This one doesn't have a 3' end, and so since it doesn't have that 3' end, we have to do it just a little bit differently. So I'm going to add it like this. Now it's going to have something called primase, which is like RNA polymerase, and so what that is right here is taking that and it will add a primer to the 3' end. So it's adding it right here, as it were, just like on the other side, and it does it in little fragments, and those fragments are called Okazaki fragments. It was a guy that figured this all out, and they named it after him. So these little fragments here, they just do them piece by piece, and those are called Okazaki fragments. So we have this RNA polymerase. We have a DNA polymerase, and what that does is it just covers it, and then it does the same thing as it would over here, and then it continues to make that separate strand of DNA. And then DNA ligase is over here, it's an enzyme over here, and what that one does is it combines those two strands together, just like those bonds that we were first talking about. Stage three, we're synthesizing the lagging strand. We have our primase, aka RNA polymerase. It moves on to DNA polymerase and the DNA ligase, and remember that they're all done in these short little fragments called Okazaki fragments. And so put all of that together, and we have another section of DNA that's going down that way. So after all of that is done, what we end with is 2 molecules of DNA and just some enzymes.